To begin with, we look into the vaccine targets. The coronavirus has four major proteins: the spike protein, the envelope protein, the membrane protein, and the nucleocapsid protein. The spike protein is the major surface protein that the coronavirus uses to enter human cells, which is composed of S1 and S2 domains. There is a receptor binding domain in the S1 domain, which is used by the coronavirus for binding into the ACE2 receptor on the host cell. So the S protein and the receptor binding domain are the main target for vaccine design because it facilitates the viral entry into the host cell during the infection process. The current data suggests that antibodies against this S protein are neutralizing, and so the majority of vaccine candidates currently in clinical trial target the spike protein. But we also have some vaccines in the preclinical stages which target other antigens like the nucleocapsid protein. So the next question is, can the virus mutate so that the vaccines become ineffective? The SARS-CoV-2 genome so far appears to be fairly stable and has mutated relatively slowly, especially compared with other RNA viruses like HIV and influenza. The SARS-CoV-2 virus said to have a proofreading mechanism which corrects the potentially fatal copying mistakes. So it mutates 2 to 6 times slower than the influenza. Until few days back, the only mutation that was described was the D614 strain, which was prevalent in the initial phase of the pandemic, was replaced by G614 strain without any significant change in the antigenic structure. So now we have the news about a new coronavirus variant from UK, and most countries are putting a travel ban fearing the new variant. So, is the new variant found in UK more dangerous? What we must understand is mutations are part of natural evolution of the viruses, and usually these mutations makes the virus more transmissible and not more dangerous. So, will the vaccines work with the new variant of the virus? The new variant has mutations to the spike protein, but the vaccines produce antibodies against many regions in the spike protein. So, it is unlikely that a single change would make the vaccine less effective. But over the time, when more mutations occur, the vaccines may need to be changed. Do you think safety will be compromised in the COVID vaccine race? We all know that the traditional vaccine development process is a lengthy, expensive process which may take 10 to 15 years. And the fastest developed vaccine till date is Ebola vaccine, which took almost 5 years. The COVID vaccines were developed at a pandemic speed. After the outbreak of the virus in December 2019 in Wuhan, within 1 month, that is in January 11, the Chinese authorities have published the genetic sequence of the virus in a public domain. And soon after that, the Moderna has announced their mRNA vaccine. And in March 16, that is less than 10 weeks after the first genetic sequence was released, the Moderna's mRNA vaccine entered the phase 1 clinical trial. So, how is it possible to develop a vaccine in 12 to 18 months' time without compromising the safety? There are few things to consider regarding this. One is, as we know, we had a SARS-1 and MERS virus infection in the past. The SARS-1 genome, which share around 80% sequence identity with the SARS-CoV-2, and several vaccines were being developed for this SARS and MERS virus. And the knowledge acquired through the development of the SARS-1 and MERS virus actually helped in shortening the time for the development of the SARS-CoV-2 or the COVID vaccine. And the next is, usually the clinical trials for the vaccines run in a series, taking multiple years. And in the case of this COVID vaccine, with an urgent need, multiple clinical trials were running in parallel. And next is, we have a new generation vaccine platforms like the viral vector platform and the nucleic acid platforms. And with the urgent need of the COVID-19 vaccine, there was a huge financial investment, scientific collaborations, and also faster regulatory approvals. And so all these things helped in the faster development of the COVID vaccine in 12 to 18 months time. So this is the traditional vaccine development with multiple stages running in series taking 10 to 15 years. And this is SARS-CoV-2 or the COVID vaccine development in which the multiple phases are combined or running in parallel, which reduce the time. And also the design and exploratory stages were shortened a few months with the pre-existing information from COVID-1 and MERS virus. And in the meantime, actually, the vaccine producers have started the production of vaccine in large scale even before the completion of the clinical trials at risk. All this help to reduce the developmental stages of the COVID vaccine. So most of the vaccines currently we are using are either a virus-based vaccine or a protein-based vaccine. For example, a viral inactivated vaccine which requires the virus to be grown in a biosafety level 3 lab and requires extensive safety requirements, and it will take considerable amount of time. So these older approaches are now being replaced by new highly flexible vaccine platforms that are basically based on the sequence information of the virus alone. So in these new generation vaccine platforms, we need only the sequence information of the virus, and we do not need to grow the virus in the lab. So in the newer generation platforms, we have the viral vector vaccines, nucleic acid based vaccines, and even the antigen presenting cells vaccine, which are in the pipeline. So these newer generation vaccine platforms are highly adaptable and speeds up the vaccine development process. So we have at least 8 types of vaccines being tested. In the virus vaccines, we have the inactivated or the killed vaccine and the weakened or the live attenuated vaccine. And in the viral vector vaccine, we have the replicating viral vector vaccine or a non-replicating viral vector vaccine. And in nucleic acid vaccines, we have the DNA or the RNA vaccine. And in the protein-based vaccine, we have the protein subunit vaccine or a virus-like particle vaccine. So this different vaccine platforms are desirable to achieve a faster global immunization because different types of vaccines may be needed for different population groups. For example, one vaccine which can be used in an older age group or pregnancy cannot be used in another group of people. Coming to the whole virus vaccines, it can be either a live attenuated vaccine or a inactivated or killed vaccine. In the inactivated vaccine, we have a major candidate from India, which is currently in the phase 3 trial. That is the Covaxin from Bharat Biotech. And also we have the Sinovac and Sinopharm from China. These are currently being used in UAE. And in the live attenuated vaccine, we have vaccines in the preclinical stages. The major one is Codagenics in producing a vaccine in association with the Serum Institute of India, and another one from Indian Immunologicals. Coming to the protein based vaccines, this can be either a protein subunit vaccine or a virus like particle. As we all know, the recombinant subunit vaccines combined with the adjuvants are relatively cheap to make and are reasonably stable, and we have a widespread experience with these type of vaccines, and the supply chains are actually well established. So if we get a good recombinant subunit vaccine, that will be a game changer in the fight against COVID. But development of this recombinant vaccine depends on the biological process. So these are slower to manufacture and develop. So these are currently running about 6 months behind the newer platforms like adeno and mRNA vaccines. In the protein subunit vaccine, we have a candidate from India, that is the Novavax from Serum Institute of India, that is currently in phase 2 clinical trials. Other protein subunit vaccines in the pipeline are a biological E and the Chinese Academy of Sciences vaccine. Coming to the viral vector vaccine, here the target gene for the pathogen is inserted into a different harmless virus using a recombinant DNA technique. So this Harmless virus acts as a platform or vector to deliver the target gene to the host cell, which in turn triggers an immune response. So, in the viral vector vaccine for COVID, the spike protein gene is taken from the coronavirus and inserted into a vector virus that is weakened so that it cannot grow inside the human body after injection. This viral vector vaccine is then inserted into the human cell, and this vector delivers the genetic code into the cell, which causes the cell to produce the spike protein. The spike protein is as such harmless, but it will trigger the body to mount an immune response. The vector virus or the harmless virus can be either a replicating virus or a non-replicating virus. So these are two types of vector vaccines: the replicating viral vector vaccines and the non-replicating viral vector vaccines. One viral vector vaccine that is currently licensed and in use is the vaccine against Ebola virus. It is named Ervebo vaccine and it is based on the vesicular stomatitis virus. This is said to have 97.5% efficacy against the Ebola virus. One issue with this viral vector vaccines is the pre-existing immunity to the vector virus. This can reduce the immunological potency of the vaccine. To overcome this, we use viruses with minimal seroprevalence in humans like the simian adenovirus and human adenovirus with less prevalence. Examples are the chimpanzee adenovirus and human adenovirus 26 and 5. Another example is the measles vectored vaccines, which also does not get impacted by the pre-existing immunity. Coming to the two types of viral vector vaccines, the replicating viral vector will replicate or produce extra copies of themselves in the human cells. One example of the COVID replicating viral vector vaccine in the pipeline is Institute Pasteur vaccine, which uses the measles vector, and another one in the preclinical stage is the vesicular stomatitis virus based vaccine. Regarding the non-replicating viral vector, this viral vector initially enters the cell and produces the vaccine antigen, but no new virus particles will be formed. The highly anticipated vaccine in India, the Oxford AstraZeneca vaccine, named Covishield, is a non replicating viral vector vaccine based on a chimpanzee adenovirus. Other examples are the CanSino Biologicals vaccine, and another one is Sputnik from the Russian company Gamaleya, which is also doing a phase 3 trials in India. Then, coming to the nucleic acid vaccines. Here, instead of a virus, a protein, or a vector, the nucleic acid coding for the target antigen itself is injected. It can be a DNA plasmid vaccine or an mRNA vaccine. The mRNA needs to reach the cytoplasm of the host cell while the DNA needs to enter the nucleus. Then this genetic material gets taken up by the cell's machinery and the cell expresses the spike protein. These spike proteins are then recognized by the immune system, which stimulates a protective antibody response. This is a new technology, and so far we don't have any other vaccines using this nucleic acid technology. Coming to the mRNA vaccine, the most discussed vaccine these days. Here, the mRNA coding for the receptor binding domain of the spike protein is used. When we say an mRNA vaccine, we must understand that we cannot use a naked mRNA as a vaccine because naked mRNA is very fragile and it is easily vulnerable to degradation by enzymes and it cannot cross the cell membranes. So, we are using a lipid nanoparticle covering for the mRNA for using it as a vaccine. So, this inherent instability of this mRNA is the reason why these mRNA vaccines need to be kept in ultra cold temperatures like minus 70 degrees Celsius. So, this mRNA encapsulated in the lipid nanoparticle is injected to the cell where the mRNA is released, and the cell's ribosomes will read the mRNA and produce the spike protein. So, to build an mRNA vaccine, we need only access to the genetic sequence of SARS-CoV-2. As we all know, the Pfizer and Moderna vaccines are the front-runner mRNA vaccines, but we also have a mRNA vaccine candidate from India, that is HGCO19 vaccine by Genova company, a Pune-based company, and it is currently in currently going to start the phase 2 trials. When coming to the DNA vaccines, these are made up of small strands of DNA encoding the spike protein and is attached to a plasmid for delivery into the body. The plasmid is used so that the body does not degrade the foreign gene before it can provoke an immune response. So here the carrier plasmid with the DNA is injected to the cell where it is transcribed to mRNA, which is then translated to the protein, which elicits an antibody response. So in the DNA vaccines, we have one major vaccines coming from India, that is the ZyCoV-D from Zydus Cadila, which completed the phase 2 trials and waiting for the phase 3 trials. And another vaccine is by Innovio Pharma USA. It is also developing a DNA vaccine. Here, there, the phase 2 trials are just started. Other major pharmas like Cadila and Sanofi are also working on DNA vaccines, but most of them have not reached clinical trials. This plasmid DNA vaccine platform is also known to show much more improved vaccine stability and a lower cold chain requirement than the mRNA vaccines. These are the frontrunner COVID-19 vaccines currently under development or trials in India. The main one is the Covishield, the AstraZeneca vaccine, which is manufactured by Serum Institute of India, and then Covaxin, the inactivated vaccine by Bharat Biotech, and then ZyCoV-D, the DNA vaccine by Cadila, then the Russian vaccine Sputnik V, which is trialed and manufactured by Reddy's lab in India. So we have around 200 vaccines in the clinical trials around the world, with at least 8 completed the phase 3 trials and got emergency use approvals in different parts of the world. So the next question is, which COVID vaccine will be India's first? So we have the Covishield and Sputnik waiting for approval from India, and the Chinese vaccines and mRNA vaccines, Pfizer and Moderna doesn't have any Indian trials. So the Indian vaccines, Covishield, Sputnik, will be the most probable candidates with an efficacy of 70 to 90 percentage, and both having a storage requirement of fully 2 to 8 degrees Celsius. Even though the efficacy is said to be 70 to 90 percentage, the efficacy against severe disease is almost 100 percentage. Now we'll go to the individual vaccines. All top candidate vaccines are for intramuscular injection, and most are designed for a two-dose schedule, except very few. Each leading COVID-19 vaccine comes with a different storage requirement. And regarding the Oxford AstraZeneca vaccine, it is a non-replicating viral vector vaccine and probably the leading candidate in India. And the main advantage is that this can be stored in a normal refrigerator, which makes rapid distribution more feasible than the other mRNA vaccines. The first phase 3 results of the Oxford AstraZeneca vaccine was announced in November 23rd with an average efficacy of 70 percentage, and a very interesting finding. That is, when the vaccine was given two full doses, it showed an efficacy of 62 percentage, but a higher 90 percentage efficacy when the first dose was a half strength dose. So now AstraZeneca is planning to conduct a global trial to compare the two dosing regimens. Either a full dose is better, or a half dose, full dose is better. The company said that the half dose, full dose regime was a mistake in the trial, but they claims that it could end up being a quite useful mistake. The next important vaccine is Sputnik vaccine developed by Gamaleya, a Russian company. This is basically a human adenoviral vector vaccine. The main difference is the two doses of the vaccine are developed with the two different human adenoviral vectors. The component 1 or the first dose consists of a recombinant adenovirus vector based on human adenovirus type 26, whereas the second vaccination or the second dose, which is taken after 21 days, is based on a human adenovirus type 5. They have used a different viral vector for the second dose, probably to avoid the anti-vector immunity or antibody that can affect the overall immunity. And the storage requirement is minus 20 degrees Celsius, but the company claims the lyophilized form can be stored at 2 to 8 degrees Celsius. And overall efficacy of the vaccine is 91.4 percentage. And in India, Dr. Reddy's and Sputnik are jointly conducting the phase 2 and 3 trials. The next important vaccine in the Indian context is obviously the Covaxin by Bharat Biotech. It is an inactivated vaccine with a novel adjuvant, a modified aluminum hydroxide adjuvant called alhydroxyl. The company claims that this adjuvant will stimulate both antibody as well as the cell-mediated response, unlike an aluminum hydroxide adjuvant which stimulate only one type of response. So this is the first homegrown COVID-19 vaccine candidate, and it was developed by Bharat Biotech in collaboration with the ICMR and is currently in phase 3 trials. This is also having a two-dose schedule given 28 days apart. Coming to the mRNA vaccines, Pfizer and the Moderna both have a reported efficacy around 95%, and the dosage is two dose intramuscular, where Moderna has a 28 days gap and the Pfizer has a 21 days gap. Regarding the storage and distribution network, this Moderna vaccine has a upper hand over this Pfizer because the company claims the vaccine remains stable in minus 20 degrees Celsius for up to 6 months, whereas the Pfizer vaccine requires minus 70 degrees Celsius. Both the mRNA vaccines don't have any Indian trials, and probably that's why the Pfizer didn't get an emergency use authorization in India. The next main vaccine is the ZyCoV-D from Zydus Cadila, an Indian company, and this is a plasmid DNA vaccine. And this is currently in the third phase of human trials, and the results we are awaiting in April 2021. And the company claims improved vaccine stability and lower cold chain requirement comparing to the mRNA vaccines. And this vaccine is being tried in three-dose schedule. Both the IM and intradermal schedule are being. The next is Novavax, which is a protein subunit vaccine. This is a full-length recombinant SARS-CoV-2 glycoprotein nanoparticle vaccine with a novel adjuvant called Matrix-M. And Novavax and Serum Institute of India has agreed for a partnership and clinical development, and they are uh, giving the commercial name as Covovax. And this is currently in phase 2-3 enrollment, and the dosage is 0 and 21 days 2 dose IM, and the liquid formulation is said to be stable at 2 to 8 degrees Celsius. So how long will the vaccine induced immunity last? Now we have a data up to around 4 months, and high levels of neutralizing antibodies are can disable the virus up to 4 months after vaccination, but we don't have a clear data how long will the immunity last. Can pregnant women and kids take the vaccine? As on today, there is no data yet on how the vaccine works in children or pregnant women, and pregnant women and kids were excluded from most of the trials. So, what are the challenges in the COVID vaccination? The main one will be the challenges in terms of the cold chain, storage, stability, and distribution of the vaccines. And next will be the initial demand for the vaccine, which can outstrip the supply because every country in the planet will need this vaccine at the same time to protect the most vulnerable populations. The next important question is, will the vaccines prevent disease transmission? Most of the COVID vaccines in the clinical trial show promising results in providing immunity against a symptomatic and severe disease requiring hospitalization, but they may not producing a sterilizing immunity. Sterilizing immunity means that the immune system is able to stop the pathogen from replicating within your body. So, if the vaccine is not providing a sterilizing immunity, asymptomatic infection can occur among vaccinated individuals. Such people may spread the disease while socializing because they may ignore the precautionary measures, believing they are immune due to a vaccine. So, the importance of the preventive measures like social distancing and mask should be emphasized till we get enough data regarding the ability of the vaccines to prevent disease transmission. Regarding the safety of the COVID vaccines, as of now, we don't have any reports of serious side effects following the COVID vaccines, and only minor side effects has been reported. But we don't have a long-term follow-up data, especially with regard to the newer platform vaccines. COVID vaccines allow a return to the pre-COVID-19 normalcy. It is important to communicate to the public that the first generation COVID vaccines are only one of the tool in preventing the COVID pandemic. Other precautionary measures should be followed. Social distancing will be a reality for the foreseeable future, and we will not be out of masks for a long time. To conclude, combined with better therapies, masks, and social distancing, vaccines could end the pandemic next year. Many questions still remain unanswered.